Right, so as this says here, the inheritance patterns that we're going to talk about next, they are known as non-Mendelian inheritance patterns, and they do not follow Mendel's first principle in some capacity. Each of these is going to work a little bit differently, okay? And so as we go through this particular lecture, I'm going to work some problems of each type, and you're going to see that you have to write the alleles differently for each type. So here we go, okay? So the first one is called incomplete dominance, right? So by definition, incomplete dominance is an inheritance pattern. Sorry, I just was refocusing a little bit there. An inheritance pattern where one allele is not completely dominant over the other allele, right? And so the heterozygous condition is going to produce an intermediate phenotype. It's going to be somewhere between the two homozygous conditions, all right? So anytime we write alleles for incomplete dominance, we use two different capital letters, right? So in this particular example, in snapdragons, which are a type of flower, if you cross a red snapdragon that's homozygous, and we can just use R's for that, with a white snapdragon that's also homozygous, and we'll use W's, then you get a pink snapdragon that's going to be heterozygous, okay, with an RW genotype, right? Now, we could put the W's in front of the R there. That's kind of irrelevant, okay? So then let's work this problem that's shown here in pink. So it says, what would happen if you cross two pink flowers, right? So over here, we're going to just go with our basic setup. I'm not going to write out all the steps just for sake of argument to make this go faster, but you would write your cross. Let me at least start with that, sorry. There's your cross of your two parents. Right, and then the alleles, you would draw those little diagonal lines. Those alleles would separate during meiosis, and then this would be in each of the possible gametes that we would write above and next to the Punnett square, okay, to be the parents' alleles and the gametes. Remember, two of these would be sperm with an R allele, two would be sperm with a W allele, okay, and then same thing for the 4 cells that are generated from the female. 2 of those cells will have an R allele, and then 2 will have a W allele, and then whichever one is the egg would be the one that's, getting fertilized, and the other 3 are polar bodies, okay? So we're going to get these genotypes that are possible in the offspring, okay? So then our genotypic ratio is going to be 1 that's homozygous for the R's, 2 that are heterozygous, to 1 that's homozygous for the W's, to be then, when we go to the phenotypic ratio, 1 red to 2 pink to 1 white, okay? So I always say, with this example, think of paint. You mix red paint and white paint, you get pink paint. It's an intermediate third, totally separate phenotype for the heterozygous individuals, right? And so not to be confused with the next one, which is called codominance, okay? So that's incomplete dominance. You get an intermediate phenotype. Remember, the prefix co means together, okay? So this is an inheritance pattern where both alleles will be equally expressed, essentially, in the phenotype of the heterozygous organism, right? So in this type of problem, we use a capital letter with a capital superscript, okay? So if we have a black chicken, that we're going to use C for chicken and B for black, so CB, CB, and we cross that with a white chicken, okay, that's going to be CW, CW, you get a black and white speckled chicken that's going to be CB, CW in your heterozygous condition, okay? So then there's that cross written out. Again, your alleles would separate. There's the Punnett square for that. Okay, we have 4, all 4 boxes would be CB, CW, okay, 100% chance of having a black and white chicken. And you can kind of tell that that's black and white. It's kind of hard to see in this tiny little bitty picture. Okay, so then what would the genotypic and phenotypic ratios be if you crossed a black chicken with a black and white speckled chicken? Okay, so I'm going to write that down here. Whoops, no, I'm just going to write the B first. CB, CB, there's our black chicken. Okay, with our black and white chicken, all right? So then over here, okay, there's our cross. Okay, doesn't really matter. We don't know enough about these chickens for it to matter where we put the alleles for the male versus the female. So I'm just putting the speckled chicken on top and the black chicken on the side. Doesn't really matter. Okay. Here we would get CB, CB. Here CB, CW. Here CB, CB. And here CB. I just lost my train of thought. Sorry. CW. Okay. So we get, for the genotypic ratio, we get 2 that are homozygous for CB to 2 that are heterozygous for CB, CW. And that can be CW, CB. It doesn't really matter. You can switch those as long as you're consistent, okay, the whole time with how you write them. If you write the B and then the W and the heterozygous individual first, then you want to do that every time. If you wrote CW, CB, if you wrote it like this, oh, you can't even see what I'm writing, okay, then you would want to do that every time as well, okay? So then for the phenotypic ratio, you're going to get 2 black, 2 black, and white chickens, okay? And of course, both of these can reduce to 1 to 1, okay?